Quería ser más independiente, quería ser mucho más responsable y aprender qué carrera podría funcionar para mi futuro y así poder organizarme mejor en toda mi vida.
Mis papás acordamos que era muy importante para mí venir a esta experiencia, no solo para aprender, pero también me da más mundo.
Me llamó mucho la atención ver que era algo que me ayudaba a decidir qué quería hacer con mi vida, pero también era una forma de divertirme y pasar bien en general. Valencia es una ciudad muy universitaria, o sea, hay muchos jóvenes, una ciudad como tranquila.
Es una ciudad que tiene de todo, tiene un montón de actividades culturales, tienen la playa, el centro, tiene un montón de lugares donde salir, donde pasar la tarde y en general la cultura de Valencia ha sido impresionante.
Los objetivos principales del Explorer Mind es que el estudiante genere autonomía, genere independencia, es tener otra mirada de los problemas, de las situaciones y ser un agente de cambio y preguntarse a uno mismo todo el tiempo, cuestionarse para generar una solución y un desarrollo innovador.
Como experiencia para los chicos, yo creo que es el mejor destino. Todos los detalles están perfectamente pensados. Es increíble cómo las sillas, las mesas, las paredes, los televisores, las pantallas, las instalaciones me parecen increíbles. La fortaleza número uno son la gente que trabaja aquí, las personas que dirigen este hub, esta institución, y realmente nos hicieron sentir que ellos van a mantener a que nuestros hijos se sientan bien. Realmente yo creo que la universidad aquí, con su gente, con la gente que trabaja para la universidad, ha ganado.
Gracias en verdad por todo, que la persona que soy yo ahorita es gracias a ustedes, que si no fuera por el apoyo que hubiera tenido no estaría aquí parada de esta forma y que gracias.
Dear space enthusiasts, thank you all so much for joining us for this very Spacefront webinar. Welcome. For those of you who are new to Spacefront, we are proud to be the first space institute in Ecuador, part of the Universidad San Francisco de Quito. Our mission is to advance space research and exploration right here from the middle of the world, Ecuador. Today, we are very excited to have Veronica Castañeda as a host with us. Veronica is alumna of the USFQ School of Biotechnology, and she's currently pursuing her PhD at the Universidad de los Andes. She's doing a lot of great studies. She's studying regenerative medicine, stem cells, but especially bioinformatics applied to space medicine. So, hello, Berito. How are you?
Hey, Andres. Hey, Afshin. Good evening, everyone. Yeah, I'm good. I'm very excited to be here, and I'm very honored. I think space research is something that has really motivated me throughout the PhD. I think it's a great source of energy for me. So it's great. I'm glad to be here, and I think it's a very great opportunity to share with people what we've been doing and share some of the motivation that we have for space research. So thank you.
Excellent. Thank you, Veronica. So now let's introduce to our incredible speaker, Afshin Meheshti.
Yes, our special guest today is Dr. Afshin Meheshti, a world-renowned leader in space biology and biomedical research. Dr. Meheshti holds numerous key positions, including professor at the School of Medicine, director of the Space Biomedicine Program, and associate director of the McGowan Institute of Regenerative Medicine at the University of Pittsburgh. In addition, he spearheads international initiatives, such as serving as president of the COVID-19 International Research Team, and that's just the beginning of his extraordinary contributions. Welcome, Afshin.
Welcome, Afshin. Very happy to have you here.
Yeah, thanks. Happy to be here.
Excellent. So let's make just one question before we dive into your presentation. I would love to ask Afshin, when you were a kid, did you always wanted to be involved in science, or you were dreaming about becoming an astronaut?
Yeah, I mean, I wasn't dreaming to become an astronaut, but I did want to go in science. I mean, I was, as a kid, I did these like fun, well, even before I knew I was a scientist, like I collected insects, put them in a jar, and see who survived first. And that was kind of a science experiment. See, it's survival of the fittest, right?
Exactly. Yeah.
So I was doing experiment, had a little microscope as a kid, putting stuff under there. So yeah, it was always science was always an interest. Yeah, and then not necessarily an astronaut, but I'll go up if someone wants to invite me to go up.
Excellent, crazy. So you are looking first to the very small things, and then you are looking up to the great things. Okay, that's
Yeah,
great,
yeah.
Action. So we are here to hear you. Thank you very much.
Great. All right, let me share my screen then. All right, let's see. Okay, I think there you go. Okay, perfect. So let me go full screen mode. Perfect. Okay, thanks. All right, so you feel free to cut me off if I go too long, but I'll keep track of the time. So yeah, thanks for the kind introduction, Veronica and Andres. Thanks for inviting me to speak with everyone here. So let's just dive into it. Why care about space and research in space? I'm sure a lot of you might already know, but there's the future explorations that are happening. There's right now a lot of the missions are happening at the International Space Station, as we know. I should have put a picture on here, but just yesterday there was a launch of the commercial missions, which I'll get into. But there's a launch of Polaris Dawn that happened, and the latest private civilian commercial mission that some of our colleagues like Chris Mason is collecting a bunch of samples and going to get a bunch of data out soon from that. And then, but eventually you want to go to the moon and then Mars, and then of course the future sci-fi idea of us floating around the galaxy and exploring it, right? But why should we care about that? Because this might not happen in our lifetime. But space. The key thing is to remember space is an accelerated model for aging, in a sense that we don't age faster, but there's a lot of health risks that are associated with aging that is accelerated in space, like mitochondrial diseases and other type of diseases. Just a little, idea of what's being launched into space. You can see the past, since the beginning, 1960s, when the first things were being launched to space until now. Really, the past 5-10 years is this big explosion of things being launched into space, from humans to all sorts of things, right? So, and this is going to keep going up now since all the commercial entities are involved. And of course, history of space travel, there's been a lot of different shuttles and things that were being done. Initially, of course, back then it was just a two-country space race, right? But now, as of course everyone has SpaceX, Blue Origin, Virgin, all these commercial entities. Of course, all the governmental entities are involved. So that's the key part where, the future of space race is really not just between countries. It's gonna, as you're gonna see, there's a bunch of data we're going to show from commercial missions that have happened. And this is sort of like the planned missions that are here in the next 20, 40 years from government agencies, NASA, or non-government agencies. You can see there's a bunch of missions that are planned. Of course, the next step is having a little moon base. Not sure if it's going to look like that, but this one graphical representation from ESA says it might look like this. It'll be nice. And then, of course, eventually you go into Mars, right? That's the next part, which might happen in the next 10 years or so. So, as probably a lot of you already know, there's, NASA has defined these 5 hazards that are associated with space flight. So why, we're going to fly in space, is it safe for humans to travel in space? This is now the hazards that we're going to face, right? So we got, distance from Earth. Of course, you can't just run down to the emergency room if something happens. You're going to have to figure out how to take care of yourself and healthcare resources there in that confined area you're in, which goes to confinement. That which leads to like fitness and other aspects, because you're just in a closed tube the whole time. And being in that closed tube, how you can have a hostile environment. I kind of think there should only be 4 hazards, and combine confinement with the hostile environment, because I think it's sort of the same thing. But these hostile environment and these confined areas, they have basically things that would evolve like infections. Like you see, for example, ISS was clean when it was first open, but us dirty humans put our microbes all over. And now there's microbes and biofilm of bacteria growing on the walls, where people have swabbed the space station in the past. Like, and probably people have seen papers on this where for example, there's microbes growing. These microbes get some antibiotic kind of resistance happening due to the space environment, the microgravity and space radiation they're exposed to. And then I just alluded to the 2 main things I focus on is the lack of gravity or microgravity, which causes a bunch of health risks, and radiation. And of course, in space, you cannot split these things up. It's the synergy of the 2 that are causing all the health risks. And of course, the job of us scientists is to figure out what's happening, what are the mechanisms. So there, there's lots of different data that's being generated by, and basically environmental things on the stations and things that happen. There's astronaut monitoring, and there's experiments that happen with different organisms, and then it's ground simulating experiments, which we'll get into. And then, you use mathematical tools or data analysis tools to kind of predict what's happening. And the eventual goal is, can you come up with some countermeasures, right, to make it safe for humans to travel in space? And the ultimate goal is like the sci-fi, you take your magic pill and now you've stopped all the damage caused by radiation and stuff. Of course, that might not happen for a while, but you never know. So, space environment, you got, as I said, I'm going to focus on the radiation and the microgravity. And, there's two types of radiation that I'm going to get into that are their main two types. Is like the solar particle events, which happens not as much, but, it's basically a solar flare, a large acute dose of protons at different energies, which the astronauts has to be exposed to. Luckily for those, you can predict when they're coming. So there might be some other kinds of things you can try to prepare for when you see a solar flare happen, or a solar particle event. Now, the background radiation in deep space is the galactic cosmic rays, which maybe a lot of you are familiar with. This is just the background radiation in space, which I'm going to go into in a second. And of course, it's the lack of gravity, which is going to cause a bunch of health risks. So I stole these slides from a colleague, which a lot of you know, Ben Walsh and Keith Sue, about this. But the fantastic forethought about galactic cosmic rays, but maybe their version might not be as accurate. They said we should develop superpowers. So, little background radiation. So before we get into it, so we refer to low LET or high LET. Like this LET is a linear energy transfer, which is the amount of energy an ion transfers to your cells. So on Earth, we're experiencing these low LET radiation, which is gamma, X-rays. So this is basically think of sand particles that travel really fast through your cells, and they'll start depositing energy as they travel through your cells. So the energy deposit or the damage will happen, but it's at a low rate. Now, high LET is the radiation we see in space. So now think of these as slower moving particles, but these are particles coming from like anywhere from protons, helium, hydrogen, to carbon, iron particles is usually between here, and then sometimes you see some sprinkling of bigger particles. So you could think of these as these bowling balls that travel slower, but then they deposit all the energy in the cell. So it's going to cause a lot more damage. So this little chart down here is showing, comparisons from the green, what we're exposed to on Earth, the blue, what kind of doses they got in space. For example, this dose here is 1 year of background radiation on the Earth's surface on average for the low LET gamma radiation. This was the shuttle mission that happened for a period of time. What was this shuttle mission 41C, which for this period of time in low Earth orbit, they experienced the equivalent dose. But remember, this is high LET radiation. So this dose is actually going to be a lot more damaging due to the amount of energy transfer to your cells. So it's going to be like 5 to 10 times more damaging, even though the dose is the same, because of the ions that are different. One thing I forgot to mention, which is important, let me go back here. Most of the data that's being produced, is in low Earth orbit. All the data that's being produced in space. In low Earth orbit, or nowadays, Inspiration 4 was a little, like if this is the International Space Station, also Inspiration 4 is at 520 kilometers. It's about 400 kilometers. And then the Polaris Dawn mission that just happened, that's 700 kilometers. Once you get closer, this is the Earth's magnetic field. Due to the physics, you're going to lower the dose, right, than deep space. But, as you get closer to the edge of this Earth's magnetic field, the Van Allen belt, the protection reduces and you get a higher dose. So that's important to coming up. But, think about the amount of galactic cosmic rays you might be exposed to and the ones that Fantastic Four is exposed to. In reality, it would have been a very short comic book and no more Marvel would have happened. Would have been one, but basically they would end up in the hospital. So what happens, with all the exposure you might get and what kind of health risks might involve? And this is kind of a chart here showing there's female and male independent of sex and dependent of sex health risks that arise, as you might imagine. The basic gist is, your exposure to space environment causes a lot of different health risks. So that's it. It's like cellular, monofocal health risks and diseases. So, you get things like, common things as you might attribute to lack of gravity is muscle generation, right? Muscle atrophy or bone loss. But there's other things related with like liver potentially disease that could happen, or kidney issues, which you might send some of the papers that were released recently. We're discussing, which I won't go over today. But there's also things like SANS, which is space-associated neuroocular syndrome, which is the astronauts start losing their vision. People think it potentially could be due to microgravity effects like the flattening of the edema or fluid shifts, but I'm going to propose potentially it could be that, but there's probably another more driving factor I'll show you in a second, which is probably driving SANS. And of course, as you might imagine, the longer you want to do the missions like to deep space, the more potential risk these health risks will accumulate. So how do you do experiments in space? Let's see if these should, okay, there you go. They're firing up now. So, there's types that you could actually send samples to, like right now, to the International Space Station. So you got your mighty mousestronauts, as I call them. So these are the mice that were flown to space, and you can see after a while, they tend to have a good time. They don't need a little wheel. They whiz around and adapt to microgravity well. This is not the only organisms that you can send to space. People have sent lots of different ones, which I won't have time to go over because I haven't done those experiments. But people send worms, C. elegans, fruit flies, squid, even plants, microbes, anything. So there've been a lot of different types of organism species sent to space. But let's say if you don't have the funding to do this, you could actually do simulated experiments on Earth. And usually, typically, to simulate space radiation, one of the only places that truly captures the galactic cosmic ray simulated beam is at this high energy physics collider at Brookhaven National Lab, which is in Long Island, New York. And I've done many experiments, which I'll show you. But what NASA had done is set up a space radiation laboratory there in mid or early 2000s, where you take part of this beam line, and in high energy physics colliders, if you're not familiar, they can generate heavy ions like iron particles or anything in between from proton, even more massive particles too. And with that, you can simulate the ions that are in space and the energies you can get in space when astronauts expose to these ions and radiation, and then expose cells, mice, or whatever other things you want to this beamline that's here, and you could then simulate the space radiation that's there. So if you want to simulate microgravity, you could never truly simulate microgravity on Earth, but you try to do as well as you can. And some experiments that could get sort of a microgravity simulated partially, or you get some effects happening with microgravity. Is one is you could do these high limb unloading. It's kind of a silly experiment. From my experience, you hang the mouse by the tail, so it's basically simulating the unloading of the weight from the back of the muscles and the fluid shifts they might get. Of course, it's not true microgravity, but you get closer to that effect. Like some collaborators of ours, Stuart Rutko and Mary Truel, like they've developed a partial weight-bearing rat model, which is a little harness there where you could actually then simulate, let's say, partial gravity, let's say on the moon or Mars too. So there's these type of models that people do. There's other types of models too, but for cells, which I don't show here, the people have, I'm sure many people are familiar here with, they have these clinostats or rotating well vessel machines that would, twirl around like this randomly to basically display the randomness of the gravity, and basically then when the rotating well vessels, how it rotates would basically simulate or try to displace the gravity there, so you have a microgravity event in quotes. So those are the ways they do it in cells. Now, if you don't have resources to do any of these experiments, of course you could. NASA set up this nice little platform of free data. Initially, it was GeneLab, but now it's Open Science Data Repository. But this is where all the omics data was initially set up, and now it's still here of omics data, where which I'm sure Andres and Veronica, our hosts, have mined very nicely for many papers, and many people here probably have, and others. But this has basically, a lot of omics type data that you could mine and tackle. And if you want from samples sent in space, from simulated experiments, and further, when this was released a couple of years ago, where now it's just not omics, but they have like all other sets of data like microscopy or tons of other types of data that you could look at. But this is a good tool for you to generate hypothesis and, let's say, test your favorite pathway that is being disrupted in certain diseases on Earth. Does that happen in space? So you could actually go mine this data and then come up with neat, actually even papers. And there's also the resources here, which you could during this time when this was starting to get developed, I forget how many years now, 7 years ago. We set up these things called analysis working groups, and I lead the multi-omics. Initially, it was these 4 analysis working groups, but now there's a lot more set up. But it's basically now it's open to the scientific community to join, and there's a lot of nice, beautiful papers that come out of this, high impact papers. And basically, it's people around the world that get together that want to work on science, space science in particular, that might not have the background. They have some do, some don't, but it's a good way to dive headfirst into space biology research. So if you want to really read about this, first you might want to look at 4 years ago, me and Chris Mason and Susan Bailey put out this big package of papers in Cell Press. And this is one of the things that the analysis working groups I briefly talked about, came with a nice handback paper. There's a lot of papers there. But now more recently, on June 11th, me and Chris worked with Nature Portfolio this time, and there's probably some of our viewers have papers in this, that put up, we put out this basically Space-omics Medical Atlas paper package. And it got nice press. And then basically now this was this package was based on this all civilians first commercial flight that ever happened, was this Inspiration 4, as many of you are probably familiar with. And what Chris had done, along with some other collaborators and PIs, they've actually then comprehensively collected a bunch of data on them, from urine to blood to swabs and other kinds of physiological type data or other kind of measurements they did with like watches, Apple Watches, things like that. But there was thousands of data points they collected, and the science got produced in here is in this package of papers that are pretty comprehensive. So it produced a lot of novel and amazing science, and now I guess Chris is really the new one for Polaris Dawn, which you guys stay tuned for that. But this gives you kind of an overview of the 44 papers, give a very nice overview of some more advanced and things, health risks that might be happening and potential ways we can mitigate the damage. I'll talk about a little bit of some things from this. So here's just a few of the papers. The ones highlighted or bordered in red are some of the papers that were resulted from the AWG team. The NASA is working on, at least for my multi-omics. So you can see that people from the group actually work together, and this is the only way really science should happen. The large collaborative groups putting a lot of nice important papers. And we ended up getting the cover of this too, which is really nice in Nature. And this is the umbrella paper, which has also WG members. But this is kind of the paper that, if you want to start somewhere in this package, read this one, because this is nicely ties all 44 papers together and discusses, kind of the overall view of what each paper has a piece of the puzzle of the health risks. But what this package does nicely is put this piece of the puzzle together as a system as a whole. And this is what this paper here describes what happens as a mechanism, cellular to the tissue to the whole body. And all these papers, when you actually see it together, you can see maybe all the overall health risks in a systematic way of things that might be impacted by space, which could help potential countermeasure development, which some of the papers in this package actually did, which I'll talk about. And there's now, as Veronica said, as of 2 weeks ago, I've started at University of Pittsburgh. We're starting a space program there, a space biomedicine program. You could actually, this Pitt Space just launched yesterday. You go to this website here, space, and there's going to be 3 pillars here, or there are 3 pillars. One is space engineering that Pitt is very strong at, and University of Pittsburgh is space science basics, and I'll be in the space biomedicine. And just to give you an idea of what we're trying to do is, and the reason I'm showing this here, because just like the AWGs, just like how open collaborative thing I've done, the whole goal of this is not only to start educational programs at the University of Pittsburgh, start programs so people can actually have a degree in space biomedicine there. Like, for example, space engineering, this semester, they've actually first started offering the intro to space engineering. So this class actually got packed as a waiting list for that. So the goal is for these two little pillars here, space biomedicine, space science, same idea. And eventually the goal is too, which I'm starting to initiate, anyone watching from any universities or institutions from around the world, including the US and international, we want to partner with various universities so we could have, for example, cross-educational programs, cross institutions. That's one thing we want to do. I mean, it's not limited to, but we want to cover these basic, I kind of kept it overall a grand scheme of things to cover a lot of things, but this list of topics including space biology that could easily expand to more and more things. So the whole goal is collaborations for educational outreach is key for, let's say, collaborations with everyone here. And then also eventually for research collaborations too, to establish kind of programs. Maybe we do it with with USFQ, University of San Francisco, we do it with you guys there, or other folks. That's the whole goal. And at first, education might be the way into the system for universities to start realizing that space biomedicine is key for the universities that don't realize it is. And again, as I'll show you, space biomedicine is not only for the 8 or 10, 12 astronauts that are going to space. Everything you do in space, as I'll show you, can easily be applied to the clinics. If you're able to come up with countermeasures of therapeutics that are going to be able to mitigate damage caused there for those health risks, you could easily apply it in the clinic too. So if you want to know more about Pitt Space or the Space Biomedicine Program, reach out to me if you want to join and collaborate with me and connect and start partnerships. So yeah, let's go into science. So this paper here, as I said, was one of the first papers that were from our AWG that had really good high impact. And the whole goal of this one was, when you approach the analysis program, I said, is there a master switch in space that could be dysregulated in humans that would cause all these downstream effects? There might have not been, but it turned out there was, and it was mitochondria. So just really big general overview of what mitochondria is. This is very oversimplification. As we say, batteries of the cell. It does a lot more than that, but the basic idea is, the mitochondria is what provides energy for your whole organism, right? And your cells, and it produces 90% of your body's energy. So that's very important for downstream functions of everything else, to your immune functions, circadian rhythm, so on and so on. And mitochondria are very important for organs, and the most bioenergetic organs is your heart and your brain. So that contains the most mitochondria. So if those get damaged, let's say because of space radiation or other factors, that's going to cause major cognitive issues or cardiovascular issues. And of course, all our other organs still have mitochondria is important for them. As you can imagine, as the mitochondria gets depleted or dysfunctional more and more, that could be related to mitochondrial diseases and things related to that. So one interesting fact about what can cause mitochondrial disease, we're not fully understanding that yet. There's a lot of mitochondrial diseases in the clinic that actually mirror what we see in space. So again, one of those things, if you figure out, let's say, how to mitigate some of the damage done on the mitochondrial space, you can actually apply that back to mitochondrial disease in the clinic. One interesting fact is, there's the 13 mitochondrial. This is the mitochondrial DNA here, the circular DNA, which has an inner loop and an outer loop. And there's only 13 mitochondrial DNA transcripts, but there's about 1,100 nuclear encoded DNA mitochondrial transcripts. So what's interesting is, the nuclear mitochondrial DNA is passed down both by the mother and father. So if you have any defects or good things happen, that could be done by both the mom and dad. But the mitochondrial DNA is only inherited by the mother, the maternal lineage. So nature decided a long time ago, men are useless and women are the key. But also, unfortunately, if there's any mutations that happen in the mitochondrial DNA, if it's in the mother's side, it'll be passed down to that. So that's some indication sometimes of, maybe some kind of mitochondrial disease formation, things like that. So let's say in space, if some mutations happen to your mitochondrial DNA and it's a female, and unfortunately, if they give birth to a child, then that mitochondrial DNA mutation could be passed on to them. So that's something to keep in mind, and it's important to know why we should stop, or we should understand the damage done to the mitochondria, because it could affect downstream generations, downstream of the space travel. And then if you could prevent, let's say, any kind of damage to mitochondria, that'll keep everyone healthy and happy for generations to come. So, the impact of mitochondrial dysfunction and disease on the body, you can see there's many different things that come up. And it very much parallels what I showed earlier, the health risks of space. So that's not surprising, since I'm going to show you data that, shows that mitochondria is a central dysregulator in space. So as I said, this first group of work, most of the work I'm going to show you, is done by an analysis working group. This was done by these folks at the time, 4 years ago. That part of, these folks, I think, still most of them all part of the analysis working group. Some more active than others, but they actually helped a lot in this project. And again, the analysis working group spread there. And I kind of told you the premise of what we did for the master switch. What the group did was actually go in the gene lab. There's a bunch of mouse tissues that were data omics that were done on mouse tissue that was more mice that were sent to space. So various people then put in expertise. They looked at RNA sequencing data or proteomic data to look at the proteins, or methylation data to look at epigenetic changes, or metabolomic data, look at metabolites, right? So people, with different expertise and interest, they took the data from these different organs. They even took data from cells that were flown to space, I'm only going to give you the big general overview of this because I'm going to show you data that hasn't been published and other things to get down to. You could always read the paper, the links here, the QR code to actually download the paper and read the details. But the whole, the whole climax of the story is that when people came back and looked at the pathways that are dysregulated in here, the top hit for everything was mitochondria. So in this paper, there's a lot in this paper, but the basic overview is that mitochondria is the central dysregulator, and downstream of that, we show in this paper, well, it has impact on the immune system, has impact on inflammation, it can impact your circadian rhythm, and so on. So there's a lot of different things we show downstream of that. What was really neat is that throughout collaborators, John Spacer, Scott Smith, and Sarah Zwart, they actually provided the largest amount of physiological astronaut data. There was about 59 astronaut data from 59 astronauts, which were things like vitamin D or cholesterol levels that could be associated with mitochondrial dysfunction. So in an indirect way, we were showing that mitochondrial dysfunction was occurring in the physiological from blood and urine, like oxidative stress levels, let's say, from markers in the urine. And then also through our collaborator, Chris Mason, he provided data from the NASA twin study. It's the end of one, granted, but it's kind of another confirmation that the mitochondrial dysfunction was actually happening, at least in one astronaut at the time. So overall, we know this paper was one of the first really centralized papers that showed mitochondrial dysfunction was happening. Before this, there were other papers that showed some mitochondrial dysfunction, but never to a point where systemically like this. And of course, people are probably familiar with the decadal survey. This is the National Academy of Science puts out every 10 years, hence decadal. And they put it on, space, kind of not in space science and space biology. And so this is kind of a guideline of what NASA might, it's a suggestion of what the research should be for the next 10 years, which NASA does take seriously. And within this decadal survey that came out last year, one thing they, of course, highlighted was, and they spent a lot of time on this, was about mitochondrial dysfunction, which was nice. They paid attention to what we put out, saying you should pay attention to mitochondria metabolic disorders that occur in space, which is very important. So now this is our version 2.0. We're going to we're about to submit this paper probably in a month. I think Veronica, she's involved in this paper too, and Andres was probably too. But it's a large collaborative network. But these are some of the key people driving it. With Joe Guarnieri, who's a postdoc in Doug Wallace's lab, and actually he's looking for faculty appointments. So if anyone wants to hire him, reach out to him or reach out to me, and I can get you in touch. He's a very talented scientist, young scientist that you could be of value to anyone's department. But so he's kind of driving, and Tin's lab from Negar and Fee, they actually use some nice, neat algorithms that they developed AI tools to analyze some of the data. So the analysis has really been driven by these folks here, and then Jean Kuhn and Chris's lab and Marissa have been actually providing some of the human inspiration for data that I mentioned that you're going to see. And then there's some other data from Fumi from Japan, from the JAXA data. So some of the key people, and of course Jimmy here, I won't show you this data, but he's done some really neat metabolic flux analysis, which you'll read once we submit the paper and the preprint will be built. So the idea is that all this paper now focuses on newer data. A lot of it's from the mouse data from Gene Lab. There's a lot of newer data there. But the human data that's been added now is from Chris's lab, and this data is now available on Gene Lab and OSDR. Because it's commercial, it was easily made available with the nice folks at Gene Lab to make it public, and anyone can mine this data now. So remember, Aspiration 4 is the 4 civilian astronauts that went up to 590 kilometers above elevation. And the dose they got, since, as I said, you get closer to the Earth's magnetic field, as you see here, this was the ISS, this is the Aspiration 4 crew was up there for 3 days. In those 3 days, they got equivalent to almost 8 to 9 months of radiation if you're down here. So they're getting closer to. So they got a little cooked, but let's see what happens to their biology or mitochondria. From the rest of the papers that were in this package, they show that after they come back after the 3 days, a lot of factors, 80 to 90% things come back to normal after a period of 120 days. But as I'm going to show you the mitochondria, some things did not. Some things got a little more cooked and are singed, I'll say, than others. But then there's also this JAXA data, Japanese Space Station data that Fumi had provided. I'll show you Masafumi Muratani, because he had this data. But basically, it's about 6 JAXA intercepts on the ISS for 120 days. And the amount of radiation they got was actually almost the same as these guys got. But they got more microgravity effect. And of course, there's a bunch of data from Gene Lab, as I mentioned. This is kind of ongoing. So, and what we did now, with Doug and Joe, the COVID project, which I'm not getting into, but in this other paper that is under review, we had developed a nice, really custom mitochondrial pathways that we used in this that we're now applying. And then, of course, for mitochondrial detailed pathways is also the mitochondria, which is publicly available. But the custom mitochondrial pathways, you can go to this paper and pull them out. You can always talk to us. We can give it to you. And this paper we're going to submit it. We have more. It's a little revised for space. So that's the basic background. So basically, what we've done now is did a deep dive into all the tissues we could find on GeneLab. That previously, I mean, some tissues were from our first paper, but now there's a bunch of other tissues that are from my sent to space, and then categorize them based on sensitivity for radiation sensitivity, right? The more high sensitivity to low sensitivity for radiosensitivity. And then also, you'll see we included a bunch of human data. Overall, what's interesting, I'm not going to go over the really big details, but this is the pathway analysis done. So on the custom mitochondrial pathways, and then usually the first group of pathways here are related with their oxidative phosphorylation activity, complexes 1 through 5. These are related to if people don't know, the complex 1 through 5 is really the main bioenergetic ATP production, energy production in your organs or your cells or your tissues. So it's very important. The purple means up and the green means down. So you see all these things there. I mean, when oxphos is suppressed in all this muscle activity, for example, that means, you got mitochondrial dysfunction suppression happening, basically, and the bioenergetics are heavily suppressed. So one other factor is, LAR is live animal return, meaning the animals came down, had a period of time to recover. ISS means the animals were sacrificed, the mice were sacrificed on the ISS after a period of time. And usually the average period of time is sometimes up to 30 days. Some vary, and I'm not going to go into all the details here. But the basic gist of it is that when the animals come down, after the live animal return, there's a heavy suppression happening, whether it's organs from the liver to the muscles, and it does have a radiosensitivity dependence happening there. Wait for the paper discusses. I'm not going to go into much of the details of everything here, but the idea is, again, you can see this heavy suppression that happening, whether it's the skin, the thymus, the spleen, some organs that are more radiosensitive, like the spleen, you can see the higher suppression happening even on ISS, as you can imagine, which then with downstream sacrifice consequences will be to your immune system, which these are all involved with. For example, when you get this suppression of mitochondrial complexes being suppressed, you get things like hypoxia, reactive oxygen species being produced, and you can see the upregulation of these things happening. What's interesting here now in the eyes, you see that this suppression happens in the retina and the eye and the ISS. This mouse here was a different strain, which could be strain dependence. But, as I said, the space-associated neuroocular syndrome is previously thought to be more physical activity than microgravity. We had, with Ethan leading this, we had to put a perspective on. Now there's a little sub-project going on in our analysis working group trying to get the data, more of this data comprehensively analyzed, but we think it's mitochondrial dysfunction really driving this. There are diseases in the clinic related to mitochondrial dysfunction that actually causes vision loss, like lung disease and other things. So this is parallel, kind of simulating that in a sense. So astronauts have come back, and for example, Scott Kelly was in the space for a year. He wasn't wearing glasses, but he came back started wearing glasses. So some astronauts recovered their vision, some don't. None have gone blind, so that's a good thing. But who knows? When you go a trip to Mars, that's a concern. There's a lot of research being done nowadays to figure out what the mechanisms are and what potential countermeasures we could stop that from happening, which is very important. Now, this is the Inspiration 4 data that Zhang Kun, he's a superstar. He should be probably a superhero kind of analysis he does, which he works on millions of different things, but very, very good scientist with Chris. And he basically showing this is the Inspiration 4 data. And main thing I want you to concentrate on is down here is the complex, again, oxphos activity happening, bioenergetics. The round circles are immediately coming back like 1 day after flight samples, and then long-term, meaning like up to 45 to 120 days after they come back. And you can see, the mitochondria still seems not to recover even 120 days later from the different bioenergetics we see. Granted, some things are getting less after, the immediate time point, but still, and I asked Chris, ask them if they were tired. And they probably wouldn't admit if they were fatigued or anything like that. And this is from whole blood. This is from the single cell RNA-seq analysis. You can see from the different cell components in the whole blood that even depending on the cell type, there's different types of suppression happening in oxphos. And what's even more interesting, this is your mitochondrial DNA here. So mitochondrial DNA, as I said, it can float around in your blood. And what's interesting is that if we see in flight. This is from the JAXA mission that I mentioned, 120 days they're in space. This is from the NASA Scott Kelly, Mark Kelly mission. Scott Kelly was there. And when this is comparing flight versus the conditions before flight. And what they see is that, the mitochondrial DNA in flight is upregulated. Now, Inspiration 4, he wasn't able to get any samples during flight. So all the samples were comparing flight versus this should be post-flight versus pre-flight. Sorry, this is a typo here. And then this is post-flight versus pre-flight here. And you can see now, you could ask, why is this mitochondrial DNA heavily upregulated, or the amount of mitochondrial DNA, because this is the mitochondrial copy numbers, is up during spaceflight. So the hypothesis here is that when mitochondria gets damaged, you get basically, mitochondrial DNA will be floating around in your blood. Now, if there's not mechanisms enough like mitophagy to clear it, and because we see suppression of mitophagy happening too, which I didn't show you that, but when things like that don't happen, then you can't clear out the mitochondria that's floating in your blood. So this might be the upregulation we might see, and an indicator of damage to your mitochondria when you're in space. But when they come back, now things start, let's say clearance levels of clearing out mitochondria B, things like that might get back to normal. But this here, from 3 different flight missions, from 3 different conditions in space, consistently shows that mitochondrial DNA is suppressed, which then the downstream effect is what we see here, the suppression of mitochondria, which is pretty interesting to see. Granted, originally when you had Scott as N of 1, this is N of 6, this is N of 4, but you can imagine if you keep repeating it over and over again with different conditions, different astronauts, you keep seeing the same thing, you're going to start believing that the data has to be real for what's occurring in space. So again, there's a lot more data in this paper, but for the sake of time, I'm just going to show you the highlights and then get ready, kind of get you guys excited for once we submit the paper in a month, that you guys get to read in full detail the preprint while it's under review. But the basic idea, this is kind of the nice schematic that Joe Ianieri, as I said, he's looking for a job. Make sure if you want a talented faculty in your lab or in your institution, reach out to him. But the basic idea is this: you can look on the right, that the more, as you can imagine, the more time you spend in space, the more damage mitochondria gets, right? So what happens is the oxphos becomes suppressed. You get these reactive oxygen species activated. You get this accumulation of potentially mitochondrial damage and mutations that might occur in the mitochondrial DNA. And then that would actually, consequence of that, where your bioenergetics decline, what can affect your immune dysfunction, causing suppression and then increased inflammation, causing hypoxia to happen then, and then eventually causing age-related disorders. Remember, space cellular modified aging, to things that happen, as I said, sands, eyes, brain, heart, muscle, and so on. So that's sort of the basic idea. There's a lot more details in the paper talking about TCA cycle dysregulation, other things like glycolysis, which I don't have time to go into, but this is the basic idea. We're very detailed follow-up to our first paper. So can we mitigate this type of damage caused? Let's say we still have time. Good. So the idea is that I had is, yes, we could probably potentially have maybe mitochondrial supplements, which I won't get into, that you potentially can mitigate it. Or maybe you could use things called microRNAs. So there's a bunch of work I've done previously in that first cell package of microRNAs, which I'll get into in a second. And there's a follow-up. So, and then again, the Kale survey paid attention, said, yeah, but you probably pay attention to these small things called microRNAs. So what are microRNAs? Everyone knows RNA, but microRNAs are these non-coding RNA. And as we know, RNA, let's go to this slide here. As we know here, RNA, basically translates to protein. That's a classical biology viewpoint, as we know. MicroRNAs now is one type of non-coding RNA. There's a bunch of different ones. And these RNA do not make proteins. They're very small. And they don't, due to their size, they don't travel alone either. They have this protein attached to them called the argonaut, which lets them get in and out of cells. But they also could be packed in exosomes, liposomes, and they could get it around floating in your blood and get in and out of cells. And as I said, microRNAs are very small, 22 nucleotides. And the single microRNA can actually impact hundreds of thousands of genes due to the seeding region they have, because it's a single-stranded, RNA, the 8-base seeding that binds to genes and could silence them. And then indirectly, when it silences a gene, it could upregulate other genes. So as you can imagine, there's microRNAs that are obviously good for bodily functions that you need them, as any kind of gene, RNA, proteins are. But of course, there's also many microRNAs that are known to be involved in all aspects of diseases, or in this case, insult, injury to your body for space. And not only are they found in mammals, but they're found in every living being, from plants, microbes, fish, so on and so on. And due to their size and due to their stability, it can be found floating in every fluid in your body, as I said, free floating or in the packed in the exosome. And the interesting thing is microRNAs are very highly conserved between species, especially between mice and humans. So when you do an experiment in mouse, a lot of times that can easily translate to human because the microRNA is conserved between the human and mouse, and that means the functionality is the same. So one thing I would propose is a good biomarker and therapeutic target for many diseases, in this case for space. Now for mitochondrial relevance, there are microRNAs that silence mitochondrial genes that have influence on the mitochondria that way. There's many papers there. There's also mitochondria produced by microRNAs called mitomeres. So these are microRNAs that translocate from the nucleus of the mitochondria that are referred to the mitochondria. Again, there's good mitomeres that the mitochondria produce for good mitochondrial functions. Of course, if there's damage done to the mitochondria, that will actually produce microRNAs that perpetuate this damage and cause further damage down the road in mitochondrial dysfunction. So, my background's in physics, but I'm a systems biologist. So me being a systems biologist, kind of a systems biologist view of what happens. A lot of people might say microRNAs are difficult to deal with, but I don't think so. So microRNAs, let's say classical biologist comes in, looks at it, let's look at one molecule at a time. So one microRNA at a time, this is one microRNA called Let7A, and this is all the genes that it interacts with and can suppress. So you can see this big hairball of genes. So let's say, for example, if you look at one microRNA at a time, you might say, oh, this one has decreases health risk, so it's beneficial. But let's say in a system, there's two microRNAs that have equal influence. One is a bad microRNA like this one, and one's a good one. And when you only look at those two, it can negate the overall what's happening. You say, oh, there's no change in health risk. But it's never any, most cases, it's not one or two microRNAs. In the virology world, that actually could be one microRNA, which we don't have time to get into. But in most cases, there's a group of microRNAs that tip this balance one way or the other for the health risk. So that's how we got to look up a microRNA signature associated with the disease. And this is an example from cancer. Like, in the cancer, you got the tumor and tumor microenvironment, which is important, but it's never just that your body's affected systemically, right? So what can affect your body systemically and say drive, in this case, cancer? It could be a microRNA signature associated with that disease. In space, of course, is a systemic response. So that's going to be impacted, obviously, by circulating factors. So that's what my viewpoint micro is. So we initially, again, I'm not going into the details of this paper. You could read it from 4 years ago. But initially, we identified a microRNA signature associated with spaceflight. And a component, there's about 13 of them in there. And each group out of the 13, it affects a certain organ or a certain functionality in your body. So 3 of those microRNAs in that overall 13, I said, should be impacting cardiovascular effects. So what we did initially in this paper, we looked at this microvasculature 3D humanoid structure from my collaborator Pete Grabham in Colombia. And what happens, you can see in this nice little imaging of that, zero grade, they kind of form these vessels from endothelial cells. There's no blood thrown through them or anything like that. It's just basically a microvasculature kind of structure. That's it. When you irradiate with the galactic cosmic simulated beam, they went to Brookhaven, irradiated these tissues, and the vessels collapse with the galactic cosmic. And we did half a gray, and half a gray is equivalent to a dose if you go to Mars and come back. So it's a total acute dose to say an astronaut might accumulate. Here's simulating the accumulated dose an astronaut might get over a year and a half mission to Mars back. But what was really interesting is that, when we applied the antagomeres, the 3 antagonists to the microRNA, so antagonist sequence, so that we could buy these things called antagomeres that would bind to the microRNA. So it's an antagonist sequence to the microRNA of interest. So we bought 3 antagomeres and then put them in the cell culture, irradiated it, and lo and behold, we completely mitigated the damage done. This is kind of the quantification of that. And what's in this initial paper, we, of course, you could do only so much in each paper. And in this initial paper, we didn't follow up with too much molecular understanding of it. We said, look, this is amazing. We should follow this up. So now this paper that came out again in June, now we dove into the real mechanism behind it. There's a lot of data here. I'm not going to go into details, but show you the basic overview of it. Oops, wrong way. So one basic thing is the overall, if you did RNA sequencing on these tissues before, during radiation and with and without the antagomeres. And one thing is these are PCA plots, principal component analysis. So this overall, it tells you how the overall genes cluster based on the groupings of the samples. So it tells you how closely the genes might be related. So 0 gray is the control. So you can imagine the controls should cluster together nicely, most of them. And this is the half a gray sham mice. So the radiation is distinctly different from the overall signature of genes that happen, as you would expect. But what's interesting, when you give the integumenters, start getting closer and closer down to the 0 gray sham mice, which just start maybe getting some understanding of what's happening. So what to keep in mind, it's going to be really difficult to give them some kind of treatment that actually physically block the radiation damage. That probably doesn't happen. So what we show in this paper is that we actually sped up the efficiency of the repair, sped up the efficiency of how it would actually prevent the damage downstream happening, and then it would actually cause this to mitigate the damage nicely. A lot of data here, but the take-home message from here is this part here, where one thing is we looked at 21 genes that the microRNAs actually shared. So as I mentioned, each microRNA has a certain amount of gene targets. Here we showed that 21 genes that actually, when we gave the treatment, they went back down to the 0 gray level. That meaning that basically, out of the hundreds and thousands of genes that each 3 of these microRNAs target, there's 21 common genes that were up during, or actually were suppressed, during the radiation without the treatment. But when we gave the treatment, it was went back down to the 0 gray level. So these genes must be important, shared by all these microRNAs. And this here is like the PCR validation of it. But this is called cumulative plots, which is I think the most informative plot here, which shows that as I said, the microRNAs have a gene seeding region which targets the genes. This is like the 8 base seeding region called 8-mer. They also could have, not all 8 bases can match the gene targets, can do 7 bases out of the 8, which will be a weaker binding, but still can silence the gene. So you do this analysis called cumulative plot, where you find how well does the microRNA, this is the fold changes on the genes. So the gene targets, how well do the gene targets compared to the seeding regions actually behave? So this middle line here is the genes that are not targeted by the microRNAs, those 3 microRNAs. Here are the 21 genes that are, and you can see they're significantly suppressed. This is fold change comparing the change from radiated to non-irradiated. You can see they're suppressed compared to your non-irradiated, as you expect, right? Because the microRNAs are going to be overexpressed and they're going to bind to these genes, and they've done that and suppress them. So now when you get an integument treatment, you can see that for those same genes, now it's getting closer to the baseline for genes that are not targeted, and this is not significant anymore, which you talk about in the paper. So that's interesting. Now he's showing that this is basically the rescue of these genes back to normal, which are important. We talk about the paper, some of the functionality of these genes, which don't have time to get into. But also what's important now, this is pathway analysis done on the genes. And just like those 21 genes we have here, we show that this is pathway analysis done comparing radiation versus 0 gray. This is pathway analysis done half a gray of radiation, galactic cosmic ray radiation again, with the integument treatment versus 0 gray. Now what's interesting is these are back down to the 0 gray level, meaning, and all these pathways are related with inflammation. So as we already know, radiation increases inflammation, possibly due to the mitochondria suppression. And what's interesting here is that now when you give the treatment, inflammation doesn't happen anymore, which is really important because inflammation could be a downstream effect of a lot of things that could prevent efficient repair also. And this is another more detailed immune inflation plot we looked at. There's a lot more happening in this paper, but really what basically we show is that indeed, if you give some of these microRNA inhibitors to these microbes that might be involved in spaceflight, you can heavily dysregulate, heavily mitigate the damage caused by the radiation, space radiation that happened. And what it didn't show in this paper or it didn't show in this part of the presentation, these microbes also target mitochondria and improve mitochondrial function. All these people involved with the paper. And now this other paper here is under review right now still. But we said let's try it in mice. So in the last 4 or 5 minutes, really quickly we did the experiment where we injected these integumenters on our simulated mice that were exposed to the half a gray simulated radiation. We also did a solar particle event type radiation with the hind limb setup, microgravity simulated or not. Long story short, from here with those 3 microRNAs that we inhibited in the mice, let's go here with our collaborator Peter and Vera out in Austria. Basically, we looked at markers related. The mice were sacrificed 24 hours after radiation, so it's really short term, like immediate effects. But we were looking at markers that potentially could downstream cause, let's say, high fibrosis in the heart, cardiac fibrosis. And what we show, for example, here, the two markers that with the radiation, look at the red highlights. With sham, these are mice that weren't irradiated, normal condition mice. You see, for example, TGF-beta, there's heavy suppression of that with 24 hours in cosmic ray irradiated mice in the heart. That's associated with fibrosis that persists. But with our integument treatment, which is here, it brings it back up to the sham level. So we rescued that effect. Same thing with the COL1 is a big fibrotic marker. This is the sham. Look at the integument treatment, we bring it back down to the same level. This is two examples that are pretty striking there. Something that's ongoing work, not in the paper. The TGF-beta is in the paper, so it's not a way of plotting it. But TGF-beta has been instrumental in mitochondrial function in the heart. Oh, and sorry, I'll show this part here that these 3 microRNAs are involved in mitochondrial related functions. Now, what's interesting in the ongoing work is MCU is a big player in mitochondrial calcium exchange and health in the heart. This green is your sham mice. This is now non-irradiated mice. Nothing happened to them. Happy mice. This light blue is your galactic cosmic irradiated mice, and then this is with the treatment of the integumenters. And you can see we've rescued the mitochondrial damage done to the heart due to this. Due to the radiation, brought MCU back to the levels it'll be. So if MCU is up, that's bad things can happen to mitochondrial dysfunction in the heart. Remember, heart's one of the more bioenergetic organs in your body. This is another, and MECU1 is also really MCU1. You can see it's up. When this is suppressed, again, this causes bad cardiac injury, and treatment with integumenters brings it back up and rescues the mitochondrial functions. So that's really nice to see. This is ongoing work. Again, this relates back to what we were showing earlier. People involved, all the people. And then now there's a paper we're about to submit with our collaborator, Mikhail, on University of Central Florida, where she's looking at the livers from these mice and the mitochondrial dysfunction. Again, we see, and I don't have time to go on this, but we see that the liver functions related to these mitochondria, these microRNAs, and we rescued them, and key markers related with factors like fibrosis, senescence, and inflammation in the liver. We're actually rescuing these effects with the treatment. So lots of data. I mean, we quickly went over some of these papers that are in preparation. Will have preprints soon, and so you'll find those. So basically, the idea is, these 3 microRNAs can be a potential way to mitigate the damage that radiation and microgravity causes, and then I'll restore the mitochondrial behavior back to normal. So all these ideas can actually be plug and play for different things, and I don't have time to go on this, but just to give you an idea. I don't do just space biology work, but all these ideas can be plug and play for COVID and long COVID. And long COVID is what basically is a mitochondrial disease, and the virus targets that and destroys your mitochondria, similar to space. So as I said, any kind of mitigation strategy we come up in space, we can actually apply that back to, let's say, long COVID patients, which is really nice to see, bring it back to the clinic. Cancer has similar kinds of behaviors too, same with the microRNAs. Working on that, and there's other factors here. But the bigger thing I want to talk about is, again, related with the whole the space program, but a bigger vision for the future that Chris, me, and collaborator Jonathan, UNC, and also George Church want to have this basically form a international kind of consortium, space health consortium called Center for Interplanetary Biology, Engineering, and Exploration. There's potential partners that might want to leverage with us, and we're talking with. But the thing is, center will be supported by these 6 pillars. Our vision is. One is this planetary data center, which GeneLab started does. So we want to hopefully partner with GeneLab if they're interested. But Chris has already set up stuff at Cornell with a biobank for all the, let's say, astronaut data, commercial ones he's getting, and the data centers that we could have in all these centers is actually one thing we're starting to develop. And we don't want to just stick with biology, but that's why it's engineering in here, the hardware and technology that you'll get to get to places we need, and the methods we need to do this, like let's say biomolecule mapping, things like that. The molecular library of life is one thing to think about. Let's say if you encounter newer organisms that we don't necessarily have the tools to recognize on Earth, or recognize with our standard tools what they are, and this kind of things in development to how to map these, and so on. So this is kind of the vision for the future, and of course, the more the merrier approach is always better. Anyway, thank you for listening. And reach out to me if you have any questions. There's a quick overview of everything, so there's a lot of data and analysis and ideas to absorb. And then I guess we're open for questions. I'll stop sharing.
Wow, Gafshin, well, that was amazing. Thank you very much for collating all that huge amount of information. There are some of my students also that attend to the meeting, so now they are more fascinated about mitochondria. I'm sure that they will see that there are more of the Krebs cycle than.
So we want to turn people into mitochondriacs. That's what we call ourselves. When you become obsessed with mitochondria, so everything's then mitochondria. So you're mitochondriac now.
Exactly.
There's always a little voice in my head that says, like, no, what is going on with the mitochondria? Yeah.
There you go. See, Veronica's a mitochondriac. We've already converted a while back. See, with Joseph and everyone else.
Oh yeah, no, I love seeing the graphs that you present, and I'm like, oh, that's the one that Joseph did. Like, it's so characteristic.
Yep.
Great. So we have some questions.
Okay.
So one is that that it's fascinating, however concerning, that mitochondria seems to show reduced resilience after spaceflight and upon returning to Earth. Is there any correlation between mitochondrial recovery and the age of astronauts?
So we haven't really been able to correlate that because, well, as you saw, the number of N is low. Scott Kelly at first, that was the first astronaut data we had. Like, oh, Scott Kelly, N once. So there was no, he was in his 40s and 50s when he was up there, right? So now the N is still low, though. We can't really make that assumption. And you got to keep in mind the people that are going up are pretty fit. Their mitochondrial is top notch. They're not like me or other average people. They're
Yeah.
So that's the thing. So this, unfortunately, for like the JAXA data, the six astronauts are from the Japanese space agency. They couldn't disclose like their age and sex due to like the HIPAA violations and stuff like that for identification purposes, which is fine. So we didn't get the age there. But the inspiration for the 4 astronauts were very diverse in age, like from 20 to 50, right? So again, but that's N of 1 of each time point. It didn't seem to matter for them in that case. But again, it's only 4 N of 1 of each time point. So the key is now to send more people in space to get their data.
Absolutely, yeah. No, that's part of the enthusiasm to have more people in space.
Yeah, unfortunately, I saw you a lot of health risks that happen and your mitochondrial damage. So it has to be people that might be willing to take that risk for a short amount of time just to get the data. But
yeah, no, absolutely. Berito, please.
Yeah, you know what I was thinking? I had a doubt about the antagomeres. Like, can the antagomeres reach all of the organs that are being affected by space, or like, should we need some kind of like a vehicle or some kind of, I don't know, maybe can I travel better in a vesicle to reach the organs that are being more affected by radiation?
No. So it's what I showed you there. I showed you a little snippet of the mouse experiments.
Yeah.
So they actually get in. I mean, there's even brain data we show from Jonathan shows that UNC had some, but I didn't show that either. But it was interesting. So the antagomeres that I got from this company, it's Ohm Biotech, a company out in Philadelphia, where Vino was the CEO of the company, but he designed it so there's these, basically it's lipid driven. You say your microRNAs, and then they designed the oligonucleotides of that to complement the microRNA, right? But at the end of these, they have these. They have this thing called a FANA technology, which you can read on their website. But basically, it's these lipids that allow these antagomeres to get anywhere, get into stuff, or circulate it and get stuff. So they actually, our collaborator Keith Sue and Ben Walsh out in University College London, that big, large kidney paper. They didn't put this data in there, but they looked at the kidneys from the mice that we did antagomere experiments on, and they actually saw the antagomeres in the kidneys itself.
Very cool.
They did the little staining on there. You could do the fish and do the marker and see that. Or situ hybridization, basically. But so they get to the tissues, and we see that like in the liver, like for example, the data that are quickly blurred through, so you couldn't actually see it. But the paper can be submitted soon, where we actually saw the microRNAs are tissue dependent too. Some of them are.
Yeah.
The blood they're circulating, but for example, in the liver, those microRNAs were already kind of suppressed compared to for the mice pre-radiated controls, right? But you saw the antagomeres suppress them more in the liver. So suppression even happened more to those microRNAs. So they obviously got in the liver and suppressed them even more. Which again, that helped some of the targets that they were binding to, or even less binding to these targets. So it was good. But yeah, so they get it everywhere. And I mean, they're expensive now, but maybe if more and more people start coming on the microRNA gravy train, and then he makes more money, and then they become cheaper, right?
Oh, that's fun, and it's also good. So antagomeres can also like sustain some level of radiation, and they can still, yeah. No, that's
Well, I mean, we've never done the study to like irradiate them, see how sustainable they are, but it seems like they do fine. Yeah, when they get the
yeah,
so
because you get the experiments. So
yeah. There's a lot of still experiments to do. So if people have ideas and want to collaborate, there's a lot to do, and people to provide funding. That's the key.
No, yeah, that's cool because I'm also like working with the London countermeasures, so I've been reading the microRNAs one to like try to relate them. So no, it's
Yeah.
very cool. I think I recently read the other one, the one with the that was
Yeah.
already published. Yeah.
Very interesting. Next time I will put a QR for donations around the
Yeah, that's right.
seminar. That will be amazing. Yeah,
Please.
a mitochondria pool, a helpline. Yeah, help us to
Exactly.
develop this thing. Yeah,
I like it.
that would be a good thing. Let's do a consortium. Yeah,
Hey, that's how I do.
let's do a pool. Okay, so these microRNAs are those that are coded within the mitochondrial DNA, in the mitochondrial DNA, and these antagomeres are coming inside the mitochondria.
So these microRNAs actually they're both. So these microRNAs actually, the literature has shown, can be derived from mitochondria. So they could be mitomeres, but they also could be from other tissues too. So that part is yes, they're mitomeres, and they also influence a lot of mitochondrial function, as I didn't show you, but as I hinted to. But so now the antagomeres getting into the mitochondria, I don't know. I haven't actually tested that. We know it binds to the microRNAs, but they're circulating and bind to them, or in the tissue they bind to them. Maybe they're getting the mitochondria and binding too. I mean, we have to show that, or someone has to show that. And I could answer your question.
New project.
Yeah, exactly. New grant project. You can write together.
Great. I have another question. So, if mitochondria are significantly affected during the space environment, could the protein responsible for maintaining the mitochondrial DNA and RNA, such as TFM, POLG, and others, also be impacted? It's like we have done like crystallography. We know that they are affected. Maybe there's any way to maintain it?
Yeah. So, I mean, in the big mitochondrial papers that come out, Joe and team, they've looked at the genes themselves too. So like TFAM, things like that is in there. Like we see the suppression of things that are happening in a lot of the mitochondrial genes that were listed, or you would expect. And what was the last half of that? You said
do we understood like any conformational changes due to the microgravity when they are
like the mitochondrial self?
Right. Yeah.
That I don't know. So that I mean, people have hypothesized should happen, but for people to actually do, like, as I said, crystallography or EM, or actually the newer technique, where one of my colleagues, Jonathan, the expansion microscopy could be a nice technique if you haven't heard of that. Like
crazy.
You throw an acrylamide or some kind of treatment to the tissue and it expands, let's say, a mouse heart to the size of a rat. And then you can actually see the mitochondria. It's really cool technology that he told me about he's using recently. Other people are too. But yeah, that I don't. If I had a hypothesis, I would say yes, it probably is conformational changing, but I have no evidence to support that. Yeah.
Very interesting.
It'd be cool if someone shows that. So if someone in the community here is listening and can do the experiments and show that, we have tissues. We have tissues to share.
Oh, that would be crazy. That would be amazing. Yeah. Now let's hope that this your webinar and your presentation will motivate all the community in order to dig a little more.
Again, space, there's a lot of unknown questions still, and a lot of these questions that these people are asking is good questions, but we just don't have the data for it. Someone has to come out and show it. And I'd love people to do that.
Of course.
Yeah.
Yeah, it's great. No, I love the Beat Space initiative. I'm glad that the site is up and that you can like tell people about the site right now. And I was just looking at it, and I think it's great. And I kind of wanted to know how can we, what are the kind of questions that we have been asking about, like regenerative medicine with spaceflight? Like I'm kind of trying to think about how can we merge those fields better. And some people have also been asking, like, what could be the benefits of merging those fields for people on Earth?
Mm-hmm. Yeah, so the regenerative medicine and the PIT are separate, but they also could be related. That's true.
Yeah.
So the point's out there. So I mean, definitely. I mean, I had one of the ideas is looking at regenerative medicine type of tissue. So obviously, let's say if you're long-term space now, if you get an injury, I mean, wound
Yeah.
healing is a big part of regenerative medicine, right? So that's something in space we know is impaired, wound healing, right? Because of lack of gravity, cells don't tend to like to, repair your wound as quick. So if they can figure out a mechanism behind that, or let's say something that your organ, let's say something happens, you get some damage done to your kidneys, kidney damage or something. Can you have regenerative medicine to maybe be able to repair that organ that's going to be damaged in a long-term spaceflight? So, I mean, these are the big overview things that are obviously not there yet, but that's what regenerative medicine can answer, those questions. That's one of the ideas in the space biomedicine we want to try to cover as programs to do, or even educational courses, right? So you as the regenerative medicine expert can give a class and say, this is what we know about regenerative medicine on Earth. This is the ideas we can apply to the clinic. So we want to train a whole new young generation of space biomedicine folks that can maybe come up with the better answers than we do.
Absolutely. No, I think that we are arriving to a critical mass of enthusiasm now. A lot of amazing institutions are developing their space institutes such as ours.
Yeah.
So this part about the educational programs that are going to be launched are going to be huge. So I think that it will help the interested workforce and scientific workforce to obtain and to launch many more experiments to answer these great questions.
And I think the key is that we all collaborate too. So you guys
Of
down
course.
there, yours, you have kind of cross collaborative classes you can do. A lot of things you can do virtually now, right? As we're doing this. So for example, let's say if there's a program that you have you guys have a lot of computational expertise down there and other things. So, or regenerative medicine, Veronica, and you might have knowledge that we don't have at Pitt, and vice versa. So then we have cross interdisciplinary
Absolutely.
classes online, and this is the same place just to you guys, anyone listening in the world, we could do that, right?
Absolutely.
That's the idea I want to push and see, and then, but of course, but get your universities on board first, right? And if they agree, then we could sign, the appropriate paperwork, and then we could start these kind of ideas push forward between. And then if one university, a group of universities we start getting the chain pieces together. Right now we get the network turns into a hairball network. Like Andres is the one. This is the one microRNA good one. So then he has his hairball network of other institutions or genes he's influencing to do good things.
Oh, just imagine all that. Count on us, Afshin. We are going to join that program. Thank you very much.
Yeah.
Great. So we just arrived to the end of our webinar. I'm very excited that this was the first one, and thank you very much, Afshin, for put yourself as the first volunteer to develop this initiative. And I hope that it will be enjoyable and also very motivating, and also for people that will be asking good questions about this part. They're not good questions, but every question will be amazing to be answered after. So thank you very much, Afshin.
Thank you
as well. Thank you for your time, and see you next time for the second episode of Spaceflight webinars.
Sounds good. I look forward to it. Yeah. Thanks.
Thank you very much, Afshin.
Yeah, thanks. Great.
Yeah.
Bye.
Bye.
I'll see you. Bye.